To deliver a discrete dosage form through intact skin directly into systemic circulation, that is the exact engineering objective of a transdermal delivery system. This graph maps the pharmacokinetic differences between methods. Oral dosing triggers sharp peaks and valleys in blood concentration, followed by degradation. Transdermal delivery, bypassing the gut, establishes a flat, tightly controlled baseline for up to 7 days. For the patient, this localized placement guarantees exact dosing compliance with minimal effort. And if toxicity occurs, termination is instantaneous. The device is simply removed. But while bypassing the liver and the gut is pharmacokinetically ideal, utilizing the skin as an entry point introduces a biomechanical contradiction. The very tissue used for delivery is evolutionarily optimized to prevent it. Human skin is not an absorptive organ. It is a biological armor system designed specifically to lock internal fluids in and keep foreign environmental chemicals out. Looking at the macroscopic architecture, the tissue consists of multiple distinct physical layers, beginning deep in the adipose connective tissue and rising through the highly vascularized dermis. But the ultimate bottleneck lies at the top. The stratum corneum serves as the primary barrier. Any molecule attempting to breach this shield must do so entirely without the help of active biological transport mechanisms. It must rely purely on the physics of a concentration gradient. Because the stratum corneum is so tightly sealed, a patch cannot simply hold medicine against the skin and expect absorption. The drug itself must be chemically selected and formulated specifically to overcome this dense architecture. This schematic details the brick-and-mortar layout of the stratum corneum. It is constructed from layers of dead, keratinized cells suspended within a lipid-rich, amorphous matrix. The precise rate at which any molecule traverses the structure is governed by Fick's law of diffusion, meaning absorption speed is dictated by the penetrance concentration, its partition coefficient, and the physical thickness of the membrane. To cross the tissue, a drug relies on the intercellular route, passing through complex lipid channels between cells. The alternative is the transcellular route, piercing directly through dense keratin proteins inside the cells. Because the intercellular route is the dominant pathway, a successful drug molecule must survive an alternating gauntlet of lipid and aqueous environments. This severely restricts the types of compounds that can be delivered through the skin. The biomechanics of the stratum corneum automatically disqualify the vast majority of existing pharmaceuticals from transdermal application. The first constraint is physical size. To successfully thread through the intercellular pathways, the active pharmaceutical ingredient must have a molecular weight of less than 400 Daltons. Molecules larger than this are mechanically rejected by the tissue. Next is the partition coefficient. Because the molecule must traverse both the lipid-rich mortar and the internal aqueous regions, it requires a balanced affinity for both lipophilic and hydrophilic phases. Finally, the drug must be highly potent. The daily systemic dose required to achieve a therapeutic effect must be under 20 milligrams. Any condition requiring high blood volume concentrations cannot be treated transdermally. Finding a drug that flawlessly meets this exact triad, low molecular weight, ideal partition coefficient, and extreme potency, is rare. Consequently, the burden of systemic delivery shifts away from the drug's raw chemistry and squarely onto the physical engineering of the patch itself. A modern transdermal patch is a multi-layered, engineered polymer delivery vehicle built to force these precise molecules into the body. The outermost layer is the backing membrane. This flexible, impermeable layer protects the internal formulation from environmental degradation and guarantees the drug flows in only one direction during the wear period. Beneath that lies the core drug reservoir, or polymer matrix. This is where the specific, tightly calibrated payload of the active ingredient is suspended. To secure the device, the adhesive layer serves a delicate dual purpose. It must bond to human skin under varied thermal and mechanical conditions, yet remain sufficiently permeable so it does not obstruct the diffusion of the drug. To aid absorption, formulation scientists often embed permeation enhancers into the matrix. These chemicals temporarily alter the lipid packing at the cellular head regions or hydrate the skin, increasing the tissue's permeability. None of these isolated components function effectively on their own. They must be fused into a unified architectural system to ensure that the device, and not the unpredictable human skin, dictates the exact rate of drug release. This diagram illustrates the first primary design architecture, the polymer membrane permeation-controlled system. Here, an ethylene vinyl acetate copolymer membrane acts as a strict physical gatekeeper, actively throttling the outward release of the drug from the central reservoir. This second schematic shows a different approach, the polymer matrix system. Here, there is no gatekeeper membrane. Instead, the drug is dispersed uniformly throughout a solid polymer network, relying on the polymer's structural density to regulate diffusion. Formulation scientists select between these two architectures based on the drug's exact potency and the physical risks involved. If a rate-controlling membrane accidentally ruptures, the patient suffers an immediate dose dumping, a rapid, potentially toxic release of the entire drug payload. A traditional passive transdermal patch represents a carefully calculated equilibrium between the physiochemical properties of the drug and the structural engineering of the polymer matrix. Yet the human variable remains unpredictable. Intrinsic barrier function, skin hydration, and lipid density fluctuate wildly, not just from person to person, but across different areas of the same body. Simultaneously, environmental stresses like heat, cold, sweating, and mechanical friction degrade the adhesive polymers over time. These real-world conditions limit the safe wear time of passive devices to just a few days. Relying strictly on passive diffusion caps the ceiling of transdermal technology. By requiring a molecular weight under 400 Daltons, passive delivery excludes therapeutic classes, such as proteins and macropeptides, which are physically too large for the barrier. To deliver these larger therapeutic payloads, formulation science requires abandoning passive physics and actively forcing macromolecules across the stratum corneum. This is the surface layout of an iontophoretic patch. Iontophoresis utilizes dual nodes to apply a small, continuous electrical current, actively driving charged, heavy molecules through the skin tissue. Other systems rely on direct physical disruption, utilizing patches constructed from solid silicon or hollow, drug-filled metal arrays. These microneedles are engineered with geometric precision. They are fabricated to a length that allows them to pierce only the uppermost dead layer of skin. Because the microprojection stops short of the vascularized dermis, they never stimulate the deeper nerve endings. The result is physical penetration of the barrier without inflicting pain on the patient. By shifting from passive polymer matrices to active electrical and physical microengineering, formulation science is neutralizing the stratum corneum. This transition enables the skin to function as a predictable interface for large molecule therapeutics that passive systems cannot deliver.